We have been talking about forming imines, and we spent some time last class talking about imine formation in the context of sugars and glycation, HB1AC or something like that. But I wanted to point out that imine formation is useful synthetically as well. You can take an amino group plus an aldehyde or a ketone in the lab, and you can, using catalytic acid, and usually, we will say what that is when we're drawing out a reaction. It could be like acetic acid, or it could be para-toluene sulfonic acid, or so on. But what that will do is give you the iminium ion, and then in the last step, you will remove a proton from the iminium ion to get the imine. All right, and we went through that mechanism last time. This is exactly analogous to the same reaction you might do, but instead of using an amino group, using an alcohol, again with catalytic acid. But the product from those starting materials is an acetal. Now, notice the difference between the imine and the acetal, but also notice the similarities. In the imine, you have two carbon-nitrogen bonds. In the acetal, you have two carbon-oxygen bonds, 2 bonds to a heteroatom. That was the same in the starting material too. 2 bonds to a heteroatom here, 2 bonds to a heteroatom there. But why do we have 2 of the alcohol groups attached to the acetal, whereas we only have 1 in the imine? The answer has to do with the key differences between this oxonium ion, that's an intermediate in acetal formation, versus this iminium ion, that's a key intermediate in imine formation. Notice that with oxonium, iminium. Notice that with the oxonium ion, the oxygen has a positive formal charge because it's bonded to 3 things, and there's no carbon, or there's no oxygen-hydrogen bond we can remove to get back to a neutral oxygen. So instead, the only thing we can do is have another alcohol molecule come in and attack the oxonium ion to give us the acetal. In contrast, for iminium chemistry, we can get to a neutral nitrogen simply by removing the proton from the iminium nitrogen to get to the neutral imine. So that's the key difference. But I don't want you to see these as separate reactions. They're manifestations of the same basic chemical principles. All right. Questions so far about that? Yeah?
I just want to make sure I'm understanding. So with the oxonium, the only way to get it to a neutral oxygen is to attack it. And with the iminium, we can just deprotonate.
Exactly. So for the oxonium, the only way to get to a neutral oxygen is to have another molecule of alcohol attack, whereas for this iminium, we can simply remove a proton. I'm going to point out that in both cases, we're using a primary amine and a primary alcohol. The primary alcohol part isn't necessarily important, but later on you're going to see that if we use something other than a primary amine, we'll get to this iminium ion and have trouble because there won't be a proton we can remove to get to the neutral nitrogen. Okay, so I want to show you just a couple of more things that imines can do in biology, and then an important synthetic use of amines. Sure. So the molecule I'm going to draw now is important for vision. This is 11-cis-retinal, and you can make it from vitamin A. or from beta carotene, which is why people have maybe told you in the past that carrots are good for your vision. We'll leave it to the food and nutritional science folks to tell us whether eating more carrots actually does make your vision better, or whether we actually get enough of this no matter what. But the protein so this molecule is normally an aldehyde. Notice the way that we can recognize whether an imine comes from an aldehyde or a ketone is to find the carbonyl carbon and see what it's bonded to. Here, a carbon and then a hydrogen. If we broke this bond, we would have an amino group on this side and a carbonyl on this side. All right, so this is a case where imine formation is actually good and intended. This large aldehyde or large functional group is embedded in the core of rhodopsin, which is an integral transmembrane protein. And this molecule is highly conjugated and therefore can absorb light in the visible region. The surroundings of the environment of that molecule in the interior of this protein can alter the wavelength at which this molecule absorbs light. And so some rhodopsin proteins absorb in the red, some in the green, some in the blue. And this is how we perceive color. The fact that this group, this large conjugated group, is attached to the protein via the imine linkage is important because when this molecule absorbs a photon of light, what happens is an electron gets promoted from HOMO to LUMO, or to an excited state. When you promote one electron from a bonding orbital to an antibonding orbital, you're going to weaken that bond. The reason that's important is because we have a cis double bond here. This is called 11 cis retinal because however we number this molecule, the double bond involves a carbon 11. We won't worry about the numbering scheme. But of course, you know that cis configurations have problems with steric strain. So when we absorb light and weaken one of these conjugated bonds, that allows the molecule to change its shape to isomerize to the trans stereoisomer. And that may look to you at this point like no big deal, who cares? But it turns out that we can't write and talk at the same time, can we? Rhodopsin. That change in shape from sort of a curved molecule to a more linear molecule in the core of the protein bumps some other things in the protein out of the way. And that change in shape causes some things to happen in the intracellular part of the rhodopsin. And what happens after that results in a signal that gets picked up by the optic nerve, sent to your brain, and interpreted as vision. And all of that happens tons of times per second, way faster than it took us to explain all of that. So it's pretty cool that we're using imines and conjugation to facilitate vision. Yeah?
And is color blindness then just a question of people not having the given rhodopsin receptors for whatever color?
That's a good question. The question is color blindness just not having one of the key rhodopsin molecules? That sounds reasonable to me, but I don't actually know. So I'm sorry about that. Somebody wants to look it up, they can remind us, but yeah. Now, one interesting thing is that your body can't take this molecule as it is and turn it straight back into the cis conformation. Instead, it has to hydrolyze off this imine, and then it takes the trans retinal, takes it somewhere and recycles it and isomerizes it to the cis. It takes the rhodopsin molecule and takes it somewhere else and then brings in a new 11-cis retinal molecule. So you're constantly recycling rhodopsin receptors in your rod and cone cells. And I forget what the cellular structures are called, but you're constantly having to recycle rhodopsin. Yeah?
So basically, it takes off the amine to recycle it, and then is it now an aldehyde?
Yeah, it takes off the aldehyde. So if you clip it off, what you've got, oops, whatever. What you have left is now this, right? And so you have to take this somewhere. This is embedded in the membrane, so you actually have to slough off the membrane somewhere to get a fresh molecule of the cis aldehyde, and you have to take this molecule somewhere and convert it back into the cis aldehyde. That's what's crazy to me, that process is so fast. And you can tell that you've overwhelmed it if you stare at a light long enough and then close your eyes and you can still see an after image. That's because you've temporarily overwhelmed the ability of the cells in that part of your retina to absorb new light molecules. They're still signaling that they've seen something, but you haven't recycled enough new rhodopsin molecules to see new things again. It's called photobleaching. But yeah, so shall we? Okay, there it is. Doesn't take long. Okay, so another really kind of cool example of imine chemistry in biology has to do with what your body does with amino acids when it doesn't need to make proteins. Okay, yep, there we go. So we'll call this amino acid metabolism. Here is an amino acid. You can draw it in a number of different ways, but I'll draw it like this. The R group can be any number of things. There are 20 or so amino acids that your body uses for proteins, a few others that it uses but not for proteins. But sometimes if you don't want to use the amino acid to make proteins, but instead want to metabolize the amino acid and use oxidation of reactions of that amino acid to make ATP or to make NADH that you can then convert to ATP, whatever. You would want to convert the amino acid into what's called the alpha keto acid. It's called the alpha keto acid because you've got the carbonyl carbon of the acid, and then on the alpha carbon you have a ketone. Just like this is called an alpha amino acid. So if you look at that, if I were trying to figure out how to do this reaction, I might look at that and say, well, it looks like I need to get rid of the amino group, or the ammonium group, convert it into an alcohol, and then oxidize the alcohol to a ketone. That sort of looks like what it is that I'm doing. Nevertheless, this reaction does not require an oxidation reagent. And the reason for that is that we're going to use a cofactor called pyridoxal phosphate, or PLP as it's sometimes abbreviated. And PLP is an aldehyde. What I've drawn here is an abbreviation for the phosphate group. And what we will see is that this reaction makes the alpha keto acid, but at the same time converts PLP to pyridoxamine. And this is why you don't need an oxidation reagent. Here's your amine. Here's your aldehyde. Here's a carbon-oxygen double bond. And here's your new amine. In other words, we're trading functional groups. The amine gets taken over to the PLP molecule, and somehow the carbon-oxygen double bond gets taken over to the amino acid. Somehow, right? So yes, in other classes they might tell you somehow, but in this class we will show you how to do it. And it just involves imine chemistry. So let's talk through how we would make the imine from PLP and the alpha amino acid. Now, if it's okay, and even if it's not okay, I'm still going to do it. I'm going to abbreviate the amino acid like this, and I'm going to abbreviate the aldehyde like this. Okay? So let's talk through what happens. First, we would probably need to get the conjugate base of the amino acid, just the neutral amine. Okay? Then we've talked before about imine formation. We usually protonate the carbonyl oxygen first to make it reactive enough for the neutral amine to attack. And you can do that in two separate steps. We could draw lone pair electrons on this oxygen attacking a proton of an acid to give us the protonated carbonyl compound. But there's some evidence that what I'm about to show you can happen as well. The nitrogen attacks the pi star of the carbon-oxygen pi bond. You break that pi bond, and at the same time, lone pair electrons on the oxygen pick up a proton from your catalytic acid. It's okay to draw it all in one step. This would be called a general acid catalysis. Because the acid part happens at the same time as you're attacking. What we may have shown you previously with you protonate the oxygen first and then the nitrogen attacks is called specific acid catalysis. The mechanism shown in your text actually has the amino group attacking the neutral carbonyl to give you a tetrahedral intermediate where the oxygen's negatively charged. There's not a lot of experimental evidence to support that. So I'm showing you what the data tell us. If you draw the mechanism, though, in the way the text has, I'm not going to penalize you for it. That would be insidious and sort of mean-spirited. So I'm not going to do that, so don't worry. All right. I just wanted to say a word that had Sidious in it because I'm a Star Wars nerd. Okay, so if we do that, we get to the following intermediate with a positive formal charge on the nitrogen. Remember that our task at this point, and I'm gonna talk our way through the mechanism but not draw the individual steps because you've seen it before in your notes. See previous notes. We need to do proton transfer to change who's the good leaving group. So the conjugate base of our acid would remove a proton here to give you the neutral amine. And then the acid would protonate the OH group to give us the positively charged water. And then nitrogen would kick down to give us water as a leaving group. So let me just want to be consistent. So after a few steps, we would get to the iminium ion. Which is positively charged, but we can make it neutral by having the conjugate base of our acid remove the proton from the iminium ion to give you the following imine, okay? So that's all chemistry you should have seen before. The iPad doesn't want, there we go. It's not responding as well as it used to. Okay, so what? Let me draw for you now what that imine looks like. I'm going to redraw the whole pyridoxal phosphate molecule because what happens next you really need to see the whole structure for. Okay, here is the carbon that used to be the aldehyde. And here is the nitrogen on the amino acid. Okay. With me so far? Okay. And notice that if we were to try to hydrolyze this amine, that would be just going backward. This imine, that would be going backwards through these steps. You can identify which carbon gets the carbon-oxygen double bond by just the nitrogen side, when it's hydrolyzed, is the amine. The carbon side is the ketone or the aldehyde. You can tell it's a ketone or an aldehyde by looking what that carbonyl carbon is bonded to. If a carbon and a hydrogen, that's an aldehyde. If two carbons, that would be a ketone. Okay? Now, the next step requires something of a jump in reasoning, though I think you'll be able to understand it based on what you learned about resonance and about acid-base chemistry. So I told you before that this is called, the starting material is an alpha amino acid because you've got an amino group on the alpha carbon. But I'm going to continue to call this the alpha carbon. Carbons that are next to an important functional group immediately adjacent are called the alpha carbon, and then you start numbering from there, beta, gamma, until you run out of Greek letters. Anybody else think it was funny that the next Greek letter in naming the COVID variants was nu, but nobody wanted to call it the new variant because probably Americans couldn't handle that? It's the new, well, what, of course it's the new variant, but which one is it? No, it's the new variant, right. Be a who's on first situation, so they had to skip to Omicron. Anyway, sorry, easily distracted. The alpha carbon has a proton on it, which we'll call the alpha proton. This proton, though it's on an sp3 hybridized carbon, is more acidic than you would expect because the conjugate base is stabilized by resonance. So I'm going to show some base, perhaps the conjugate base of our acid, perhaps an enzyme functional group can remove a proton from the alpha carbon. And when it does so, I will draw that conjugate base, and I want you to see and convince yourself that that conjugate base is resonance stabilized. And this will be important a lot in subsequent chapters because we're going to see a ton of times actually, I'm just going to draw this, and here is the negative charge. We're going to see a ton of times where we remove a proton from a carbon alpha to a carbonyl, and then we can do interesting things with that. All right, do you see how this negative charge is adjacent to that pi bond, but it's also adjacent to that one? So really, we have a 5-atom conjugated system here. And the negative charge could be on the oxygen, on the alpha carbon, or on this carbon from the imine. I'm going to draw those 3 resonance structures, but because I don't want to spend all the time to do it, I'm going to abbreviate, I'm going to copy and paste. I know that that's irritating to those of you who are using paper, but I apparently don't care enough to not do it. And actually, you're not going to need to copy this down. You can come back in your notes and do it later, or you can get the PDF notes from me. So you've got a resonant, sorry. You've got a resonance structure in which you can put a double bond here and then negative charge out on the carbonyl oxygen. And you have a resonance structure where you can take the negative charge here and make a double bond between carbon and nitrogen, and then move these electrons out on to this carbon. So I'm going to zoom out and show that to you again. But do you see how resonance makes it so that charge is delocalized? Yeah?
So, sorry, Professor, to take you back a little bit, looking at the general versus specific acid catalysis, and you said you won't penalize us for doing it either way.
Right.
Your tests are all multiple choice.
Right.
So what do you mean? Will there be like multiple choice questions that result, or is that?
Oh, yeah, so the question is, when I said I wouldn't penalize you, of course, if it were a free response test and you showed the mechanism in an alternative way with these proton transfers as I've discussed, I wouldn't take off points for it. But yeah, on a multiple choice test, what I'm telling you is I wouldn't give you option A, general acid, option B, specific acid, option C, what your textbook chose, and then guess which one I think is right today. Okay. I wouldn't do that, yeah. I might ask you which of these are correct more than one option. That would be kind of evil, but I wouldn't ask you to choose which of the ones, all right? Okay, so it turns out this resonance structure here on the left is going to be the important one, because if we pick up the proton that we removed at that carbon, then look at what we have done. Now, what used to be the carbonyl carbon of the PLP reagent is sp3 hybridized. It has 2 carbon hydrogen bonds and a single bond to nitrogen. Okay, and in contrast, what used to be sp3 hybridized here has 2 bonds to nitrogen. We've moved before. This carbon was the imine carbon, and this was the sp3 hybridized carbon. Now, what used to be the imine carbon is sp3 hybridized. What used to be sp3 hybridized is the carbonyl carbon, or the imine carbon. And we did that just by proton transfer and resonance structures. Okay, there was no oxidation or reduction, because once we removed this proton, the negative charge was here, here, and here. We could protonate on any of those places, and if we protonate here, we get this molecule. Now notice what I told you before about trying to decide which side of the imine is what. Just draw a squiggly line down the middle and note that this is my carbonyl carbon and this is my amino group. So now we're going to do imine hydrolysis. And again, I don't think we've necessarily walked through imine hydrolysis completely, but I'm not going to show you every step because they're just the reverse steps of what imine formation is. The first thing, we can talk it through though, the first thing you would need to do is protonate the imine nitrogen to make the iminium. Then you would have water attack. Okay, does that make sense? Then after water attacked, you would have this intermediate. and the nitrogen would no longer be positively charged. But we want water to stay and nitrogen to leave, so we need to change who's the good leaving group. We would remove a proton from water or from oxygen to make it the neutral OH group. We would then add a proton to nitrogen to turn it into a good leaving group. Then electrons would kick down from the oxygen and kick off the nitrogen. At that stage, we will have liberated the portion of our molecule formerly known as PLP. The molecule formerly known, yeah, it's difficult to say. This is pyridoxamine, which I don't expect you to know or memorize what the structure of pyridoxamine or what the name is, but you'll hear it in biochem. And so when you do, I want you to remember that you heard about it here first. Maybe not first, maybe you already knew this stuff. And then left behind is this oxonium ion, where we can use the conjugate base of our acid to remove that proton, and then we get the alpha keto acid. The carbonyl group is now on the alpha carbon of our acid. The amino group is now attached to what used to be the carbonyl carbon of PLP. Okay? Questions you want to ask about that? Yeah?
So basically, the reason we needed that specific resonance structure so that when we did amine hydrolysis, the nitrogen could stay on the molecule.
Right. Yeah, let me say that again. The reason that we did the deprotonation and reprotonation was so when we did imine hydrolysis, the nitrogen could leave with the PLP side of our molecule. Yeah.
This is a lot like the amino acid metabolism we did earlier, isn't it?
Did we do this earlier?
It just seems like the reverse reaction.
Yes. Oh, sorry. Yes, we began with amino acid metabolism. I'm sorry, the whole thing is amino acid metabolism. In other words, we made the imine first, then we did the proton transfer to switch where the imine was, and then, yes, you're exactly right, we're doing the reverse reaction where we're hydrolyzing the imine. Yep. Now, why does biology do this? Well, the answer is it's efficient because you don't have to do redox chemistry. You could presumably oxidize this to the imine and then hydrolyze the imine to liberate your alpha keto acid and ammonia. But have you ever taken a good whiff of ammonia? At our house, oh my gosh. Well, we have boys and they're smelly, but it's not actually the boys that are bad. It's the cleaning rags that get just disgusting after a while. I don't know if you've had a rag that's sat in the kitchen for a while and you smell it and it's got this nasty smell. It turns out that nasty smell doesn't necessarily go away even when you wash your rags on hot, and so we put in a little bit of ammonia and that kills whatever's making the stink, and then it's better. But the cure is almost worse than the disease because you pour ammonia dissolved in water into the washing machine, and then it's just this really sort of acrid smell. And yeah, it's toxic, it'll kill you. So your body doesn't use ammonia when it wants to transfer nitrogens. Instead, it uses imine chemistry. So think of pyridoxamine as a transfer mechanism for ammonia, because we can now take pyridoxamine and some other alpha keto acid and make a new amino acid by going backwards. Nature wants to be able to do these reactions backwards and forwards to balance the needs of the cell and to have the right amount of molecules in the right places. Yeah?
I might have just missed this, but what did you do to deprotonate the NH group to get it to the NH2? In the very first step.
Yeah, I skipped over that. Where did I deprotonate the NH3 to get to the NH2? There we go. Okay, what else? All right. I want to come back to a use of imine chemistry that's useful in the lab. If I wanted to take this primary amine and convert it into a secondary amine, you'd think that the SN2 reaction would be a nice way to go. But it turns out that the secondary amine is even more reactive than the primary amine because alkyl groups are electron donating. So the nitrogen is going to be even more nucleophilic, and as soon as I form some secondary amine, it's going to react with some of the remaining alkyl halide reagent to give me the tertiary alkyl halide, which is itself more reactive because it's got more alkyl groups. So that's going to go on to react with some of the remaining alkyl halide reagent until what I have left is the quaternary ammonium, and then I'm done. This problem is called overalkylation. So this is not a really great way to make amines. If you want to do it in a controlled way, here's what you do. You first take the primary amine and you react it with some catalytic acid and an aldehyde that has the right number of carbons. When you do that, you will make the imine, and then we're going to reduce the imine with sodium borohydride. Remember, this is a reagent that can deliver H- to ketones and aldehydes. It works for imines too via the same mechanism. We deliver the hydride to the pi star at the carbonyl carbon. We break the pi bond. That generates this negatively charged intermediate, which will pick up a proton from somewhere and definitely in aqueous workup. Not sure what, I'm not trying to communicate that we add acid. This is probably in a workup step. That will give us the desired secondary amine. And we never ever run the risk of overalkylation because once we've added our nucleophile to the imine, there's really nothing else that can happen. This process is called reductive amination. It's a two-step process. And if you want to hear the full story on this, you can go to the lecture at 9 a.m. By the full story, I mean the story of the time I tried to do this reaction in the lab in grad school. I tried to do it on a large scale, and my hubris was my downfall because the large-scale reaction bubbled over while I wasn't there, and I had to go back in the middle of the night and undertake to cover my sins by cleaning up the spilled reaction that was all over my fume hood and all over the ground in the lab. And then, because confession is good for the soul, I had to tell, I'm already telling you the story, so I guess you don't have to look it up. I had to tell my advisor, my PhD advisor, that what I had done, and then he wanted me to tell what the whole group had done so I could be a cautionary tale for others. And the moral of the story is, if you're going to do a reaction on large scale, do it on small scale first to make sure you know how to do it, which I had done. But then the other story is, when you're doing something on large scale, you can't walk away from it and expect it to all turn out well. Sometimes large scale stuff doesn't work. If you've ever been in the kitchen and tried to make like a quadruple batch of something, sometimes it doesn't go as well as the smaller batch does. I'm sure there are some gospel living applications in all of that. I will leave it to you to find them. He that hath an ear, let him hear. Okay. So in the last little bit, I want to just briefly mention what happens when you use a secondary amine in imine formation. So let's use a secondary amine and this ketone, and let's use catalytic acid. The key problem, and we'll jump ahead several steps in the mechanism, is you get to the iminium ion, and in contrast to the iminium ion you get when you use the primary amine, there is no proton to remove to get to the neutral molecule, at least not on the nitrogen. And so what happens next is, instead of trying to remove a proton on the nitrogen, we're going to instead remove the proton from the alpha carbon for reasons similar to why we removed the proton from the alpha carbon earlier today, because you're going to see that the conjugate base of the iminium ion, where we remove the alpha proton, is resonance stabilized and has a neutral form, which is the one that we will usually draw. This is called an enamine, though I think your text writes it this way. The E is long. Anyway, enamines are products when you start with a secondary amine. You don't need to think of this as a separate reaction. It's just the natural consequence of using an amine that's secondary. We may come back later on and talk about, when we start talking about doing things with deprotonated alpha carbons, we may come back to the enamine because it's actually a useful reagent in organic synthesis. Okay, so there's two more things we need to talk about. It's sort of a smorgasbord, and it's sort of related to what we've done so far. It's reactions of aldehydes and ketones. I'm not going to hold you responsible for the mechanisms of these next two reactions, though I do want you to know how to use them in a synthesis and how to predict the products and so on. I hate to just talk about reactions like, okay, here's a reaction, there you go. But, and so I would just omit these, but I'm concerned that some of you on future standardized tests like the MCAT or the DAT may encounter these reactions, and so I want you to at least have seen them. The first is taking an aldehyde or a ketone and reacting it with Cn- as a nucleophile. Usually we use sodium, the sodium salt, sodium cyanide, and we do this under aqueous conditions. AQ means aqueous, water's present, and we use HCl as a salt. You have to be careful with this. If you ever do this reaction, you need to look up a procedure for somebody who's done it previously, because if you have too much HCl, you protonate Cn-, you make HCN, which smells like almonds but is a toxic gas. And I think if you smell it, you're nearly dead, so you need to run. So don't try this at home, kids. What happens, and I won't draw the mechanism, though your text shows it and you can look it up, is Cn- attacks to give you a tetrahedral intermediate, then you protonate the carbonyl oxygen. Or alternatively, you can protonate the carbonyl oxygen first. In any case, the product has an OH group and a CN group on the same carbon. And the reason this is useful synthetically is because you just made a carbon-carbon bond. And carbon-carbon bonds allow you to make more elaborate structures. They're sometimes difficult to do, and so when you discover a new carbon-carbon bond-forming reaction in OChem, you sort of go, ooh, that's fun, and you sort of remember it. One of the useful things you can do with this product, which is called a cyanohydrin, is to use a strong acid and heat, which in OChem we usually use a delta because, I don't know, it looks like a little fire. That replaces the 3 carbon-nitrogen bonds with 3 carbon-oxygen bonds and converts the CN or nitrile group into the carboxylic acid. And the reason we're doing this is because carboxylic acids are very useful. They're easy to modify and convert into other things. So this is a useful synthetic sequence. I do not care whether you know about the mechanism of going from the nitrile to the acid, though if you want a hint in sort of SAT form, imines are to aldehydes as nitriles are to carboxylic acids. You can actually convert them using the same basic steps. If you're bored and lonely sometime, give it a try and see how far you're able to come up with. Okay, so that's one reaction. I want you to be able to use that in a synthesis. I want you to be able to predict the products, but I don't care that you know the mechanism. The second is the Wittig reaction. If you say Wittig, we will laugh at you. That's because it was named after a German, Georg Wittig, who won the Nobel Prize in 1979, which was the year of my birth, 150 years ago. May as well have been. You know, it's interesting to think about life. We don't have any time for this. In the 1800s and how similar it was to life in the 1500s, and yet there's a huge difference in how we live today versus how my parents lived when I was born. The Wittig reaction is kind of Lego chemistry. It's carbon-carbon bond formation. You take an aldehyde or a ketone and the Wittig reagent, and the Wittig reagent is weird. You won't have seen it before. It is an alkyl group that is bonded to a phosphorus. The phosphorus is bonded to 3 other groups, in this case, phenyl groups. It doesn't have to be phenyl, but that's the most common. That's the most common option. And because phosphorus is from the same row of the periodic table as nitrogen, when phosphorus is bonded to 4 things, it has a positive formal charge. And the Wittig reagent also has a negative charge. The carbon adjacent to the phosphorus has been deprotonated and has a negative charge. And what happens and we'll talk about how to make the Wittig reagent, but what happens is you replace the carbon-oxygen pi bond with a carbon-carbon pi bond. Where stereoisomers are possible, you get both. And then the other byproduct is, this is where the oxygen goes. The oxygen from the ketone goes with the phosphorus. This is triphenyl phosphine oxide. Phosphorus here is bonded to 5 things, which should make you uncomfortable, maybe give you a little bit of heartburn. But remember, one of the reasons that second row elements don't adopt more than 4 bonds is their small size. There's simply no stable way to orient 5 things around a carbon or a nitrogen and have the bonds be strong and tight. Whereas if you're bigger, as in the case of phosphorus, there's plenty of space for that kind of thing. So phosphorus being a 3rd row element, you're going to have to get used to seeing it with 5 bonds. All right. Okay. And of course, the ketone would have been this group. Now, this molecule. So effectively, you're clicking together this carbon and this carbon. And so the Wittig reaction is a great way to take simple starting materials and bring them together via a carbon-carbon bond at a late stage. What the heck am I doing? Thank you. You're like, indeed, what the heck are you doing? This carbon, yeah, is that better? I don't know what came over me. Okay. So what I will show you, just for your information, is I'll show you how we make the Wittig reagent, and then I'll show you the mechanism of this reaction so you can see once how it happens. I will not ask you to reproduce that mechanism, but I feel like this reaction's weird enough that if you don't see the mechanism, you may just forget the reaction entirely. So with that, we'll pause. We'll pick that up next time. Have a great weekend.